Good morning or good afternoon, everyone, depending on where you are. Thanks for the nice introduction, and thanks to Don Whitley for inviting me to give this webinar on how we explore the microbiome and how it interacts with the immune system. Before I start, I would like to give a land acknowledgements. The University of Calgary is located in the heart of southern Alberta. I acknowledge and pay tribute to the traditional territories of the peoples of Treaty 7, which include the Blackfoot Confederacy, comprised of the Siksika, the Piikani, and the Kainai First Nations, the Tsuut'ina First Nation, and the Stoney Nakoda, including Chiniki, Bearspaw, and Goodstoney First Nations. The city of Calgary is also home to the Métis Nation of Alberta, districts 5 and 6. So I don't think that the microbiome needs a big introduction to this audience. Just very quickly, as we all know, all of our mucosal and barrier surfaces are colonized with microbes. That starts at birth. Microbes include bacteria, viruses, fungi, and protozoa. This talk will focus on the commensal bacteria in the large intestine mainly. But as I said, we are not only colonized in the gut, all of our body surfaces are colonized as shown here. The microbial composition and diversity depends on the niche, so each site has its unique microbiome. Nevertheless, the microbiome is individualized between individuals. So if you look at the high resolution level, you have kind of a fingerprint with your microbiome and all of those body surfaces. If you focus now on the intestinal microbiome in the large intestine, we can say that only about 8 to 12 of the 55 or more currently known bacterial phyla are present in the human microbiota in the intestine, which already demonstrates that this is quite a specialized environment and that this colonization pattern or the composition really has evolved throughout evolution, and both the host and the microbiome benefit from this relationship, of course. More than 90% of the human gut microbiota at the phyla level is composed of 4 major phyla that are listed here. But we also find other phyla at a lower abundance, such as Fusobacteria and Verrucomicrobia. Now, importantly, the majority of these bacteria, commensal bacteria that are present in the large intestine, they are anaerobes because the oxygen level is really low in the gut. So if you want to culture or work with those bacteria, you need to have anaerobic capacity. And this is where Don Whitley with their anaerobic chamber comes in for our work. Now, when we zoom in though from this phyla level to the species level, it becomes clear again that all of us have kind of a fingerprint. The composition is different between all of us. So it is really important to study the function of this microbiome and, as I said, how it interacts with the immune system. Now, because the human microbiome or also the microbiome of standard laboratory-specific pathogen-free mice is so complex, this makes the study quite difficult, which is why we use gnotobiotic approaches that I'm going to talk about a little bit later. Now, why do we want to study that? What do I mean with the function of the microbiome in an immunological sense? We now know that actually colonization that occurs at birth, that then the colonization or the microbiome interaction with the host is really, really important throughout early life, as indicated here, and the first 1,000 days are an example for the human population. Because the first 1,000 days of life in humans, they represent a critical window where not only the microbiota, but for example, also the immune system that we study, but also the brain, where the brain-gut axis comes into place, develop, and all those developmental processes are coordinated, of course, and they influence each other. So we know that early life, and we know that even prenatal, so that the microbiome of the mom during pregnancy is really a period where those microbes and metabolites that come from those microbes can influence maturation and regulation of the immune system. And as I said, that is particularly permissive to education from the microbiota during early life. These immune effects or maturational effects in this critical window early in life can be or are often long-lasting, which means that this immune education during this critical window of opportunity can impact resistance or susceptibility to diseases later in life. If we want to study the specific effects of the microbiome on the immune system at specific time periods, we also have the problem of facing how to achieve temporal control over colonization in mouse experiments. And this is where we're going to get into the genetic manipulation a bit later in the presentation today. So how can we study this? These effects, it's impossible to study them in detail or at a mechanistic level in the human population. We do know that many diseases, or actually probably every disease you can find nowadays, has somehow a microbiome component. So, for example, we can see that diabetic people have an altered microbiome compared to healthy people. Now, the question is this cause or consequence? What I mean by that is the disease caused by the change in the microbiome? So is the microbiome changing first and then that's causing disease? Or is the change in the microbiome a consequence of the disease? Do you just change the microbiome because a diabetic person has a different niche or environment? And to study this, we use gnotobiotic mouse models where we can define the bacteria that are present, and we can also define the host, which allows us to perform mechanistic experiments. So how does this look like in reality? So we use axenics and gnotobiotics at the International Microbiome Center of the University of Calgary. And as you can see already, this approach requires highly specialized infrastructure, mainly isolators that are shown here, where we can house germ-free mice, so axenic mice that are completely devoid of microbes anywhere. So no bacteria in the gut, no bacteria on the skin. They are basically an empty vessel that we can use for our experiments. As you can see, we do not only need specialized infrastructure, but also highly specialized operating procedures where we need to import everything from an autoclaved drum. This includes the material, the water, the food. So the biggest challenge is really to keep those mice germ-free and avoid any microbes from coming in. Now, the next step, experimentally, of course, is to be able to test individual commensal bacteria that we can culture in pure culture and then use in experiments to colonize those germ-free mice and see what kind of effects they have. And that's called gnotobiotic, where we study organisms that are defined. So we can take 1, 2, 3, 4, 5, however many we want. As long as we know which bacteria are present, we have a gnotobiotic model. Now, this setup gives us really strong experimental control over the conditions in which we perform our experiments. By using inbred laboratory mice, we obtain control over the host genetic background. We can also use knockouts, knock-ins, reporter mice, fate mice. All those tools are available to us. Through gnotobiotics, we can control the microbes that are present and the microbial composition in the microbiota in those experiments. And lastly, what I'm going to talk a little bit about later, we also have, in addition to that, now genetic control over more and more microbes where genetic systems become available to manipulate them. Now, in addition to study individual microbes and their function, we also would like to study how does the diversity or complexity of the microbiome impact those functions. And to do this, we need to use model microbiotas, as we don't really want to go back to the completely complex and diverse human microbiota. So due to that, many model microbiotas have been developed over time. When I started working in the field 12, 13 years ago, we used the altered Schedler flora, which was developed by commercial vendors just to normalize the microbiota without any real rational background. We then were involved in developing the oligo-MM12 or SDMDM-M2 microbiota, which we often use at the moment. And that's just a composition of 12 commensal bacteria that kind of mimics the phyla composition of the human microbiota. But many groups are now expanding and making those model microbiotas more and more complex, but still defined, just to cover more and more of the metabolic functions that are actually present in the human microbiome. And here the HCOM1 and HCOM2 shown below, they are just one example of this. So as you can imagine, building those microbiotas and having all those commensal bacteria in pure culture really requires a high capacity of good anaerobic culture conditions. And the way our lab or the groups here at the University of Calgary approach this is by using the A95 large anaerobic chamber from Don Whitley. And the main reason we use this system is because it has a large import port to get materials in. And most importantly, we needed 2 people to work at the same time. So we didn't go for the large option because we needed the capacity for plates, for example, to culture in there. We really need the capacity for 2 people to work together in order to prepare materials. So in gnotobiotics, the biggest challenge that we face in experiments is contaminations of the mice or contaminations of samples with bacteria that we don't want. And since we have groups that work either with mouse microbiota or mouse anaerobes isolated from mice, but we also work with human fecal samples and isolate bacteria from those samples, we really need to keep those 2 systems separate. So at the Foothills campus, where my lab is, we actually have 2 A95 units, one for mouse anaerobes and one for human anaerobes. So in the mouse chamber, we culture anaerobes from mouse feces. We prepare anaerobic commensal bacteria or communities of bacteria for germ-free colonizations. And this process is quite, the SOP is quite complex, so you really need 2 people to work at the same time to prepare those kits. We, of course, where possible, where we have the opportunity to genetically manipulate those commensals, we also need that chamber for that. On the other hand, in the human chamber, we culture anaerobes from human feces and we process feces samples for storage or DNA isolation and processing under anaerobic conditions, just to make sure we don't lose any species. Now, this is the operational aspect of generating defined microbes or microbial communities that we then can use in gnotobiotic experiments. Now, as I mentioned before, if we want to test effects in vivo that are happening in a specific window of time, we need to have the ability of temporal control over colonization. Because naturally, if you colonize a mouse, the microbes, even if it's just one E. coli, the bacterium colonizes, reaches a high level, and then stays. And it's impossible to get rid of it, even with high levels of antibiotics. You'll never get rid of them completely again. So a system that a postdoc in Andrew McPherson's lab, where I did my postdoc in 2010, developed is an E. coli strain where we basically knocked out the enzyme that's responsible for converting L-alanine to D-alanine, which is required to build the cell wall. D-alanine is not available in the host, so all of our amino acids are L. So if the bacteria can't convert the L-alanine to D, it's unable to build its cell wall and divide. But it is alive and metabolically active as long as it doesn't try to divide. So this means that we can grow wild-type bacteria or the mutant when we supplement with D-alanine, as you can see here in vitro. But as soon as we remove the D-alanine from the medium, the mutant, the auxotrophic mutant, disappears. And as you can see, this also works in vivo. The wild-type colonizes stably, and the mutant, since D-alanine is not available, if you gavage a high dose, you reach the same levels as the wild-type, but then it drops. And repeating these procedures allows you then to time, for example, during pregnancy only, to make sure that we only have commensal bacteria present during that specific window that we want to study. Now, of course, E. coli is an anaerobe. All the tools to genetically manipulate it exist. So this is a beautiful example that this system works. And the interesting fact is also that this D-alanine is basically used in all bacteria to build their cell wall. So because Bacteroides, for example, are a much more dominant species of bacteria in the microbiota than E. coli, we wanted to adapt this system to several Bacteroides species. I apologize for the blurred data pictures, but since this is a public webinar that's going to be posted and this is unpublished data, I didn't feel comfortable showing the high quality data. But what I can tell you, and you just have to take my word for it, is that we managed to genetically modify 3 relevant Bacteroides species by knocking out the analog, so the same gene that's responsible for converting L-alanine to D-alanine. And as you can see, all 3 species, when we test the wild type, they colonize consistently. But when we use the oxotrophic knockout, we get high levels of the high dose gavage that drops to undetectable level for all 3 species. This now allows us to perform experiments with temporal control using Bacteroides species. And our goal is, of course, to also expand that to include other commensal bacteria, such as Clostridia, lactic acid bacteria, or Akkermansia species, for example. And then eventually, since all of those oxotrophic species are dependent on D-alanine, we hope that we can build actually communities of 4, 5, 6, 7 oxotrophic species, which allows us to have temporal control over colonization with a community rather than with only an individual bacterium. Now, the next step that we are doing, or the microbiome field is stepping into at the moment, is into using wild microbiome mice. And what I mean by that is we moved from complex standard SPF-specific pathogen-free laboratory mouse microbiota to gnotobiotics with defined composition, just to be able to perform mechanistic studies and have really defined experimental conditions. But on the other hand, we also know that many findings in mouse models don't translate well into clinical studies. And there are some studies out there now that show that when you use a wild microbiome from a wild mouse that has a much more significant effect on the immune system, it seems like these mouse models can predict much better whether something would work in a clinical study or not. Therefore, we are now building, or we are in process of building, have almost finished a wild microbiome center at the University of Calgary, which has to be completely off-site on another campus, because we really want to avoid contaminations with anything that comes from those wild mice. So we have in this wild microbiome center a third A95 chamber to culture commensal bacteria from those wild microbiomes. And of course, they don't only have bacteria present, we can also culture other microbes such as fungi. This allows us to either isolate those wild commensal bacteria, or we can take fecal samples from wild mice and transfer them into laboratory inbred mice and generate wildlings to study effects in the wild. So this gives us here at the University of Calgary the possibility to experimentally test effects over the whole spectrum, from germ-free mice to monocolonized gnotobiotic mice, defined microbiotas. We can double check in SPF mice to see how it fits with other literature that was done in SPF mice, and we can move our experiments into the wild to really make sure how this looks in the most physiological setting. And as you can see, for all of those steps, we rely heavily on the capacity to do efficient and clean anaerobic culture, where the A95 for us is really the go-to chamber to use for that. So that's all I wanted to talk about today. All of this would not be possible without the people that actually do the work. In my lab, I've shown data from Dr. Shukru Ahmadi, a postdoc in my lab, as well as Sharon Dong, a master student who generated one of the oxytrophic bacteria that I've shown, the Bacteroides. This is all in collaboration with Dr. Cassie McCoy, who's the lead of the International Microbiome Center and the WIMIC, the Wild Center. Dr. Paul Cubes is also the director of the Wild Center here at the University of Calgary, and Dr. Karen Thompson was also involved. We also collaborate with Dr. Philip Ahern and Dr. Mohamed Vidar from the Cleveland Clinic, who helped us generating additional oxytrophic Bacteroides species. And here I'm listing the funders that support our research. I'd like to thank Bethany Rook from Don Whitley for organizing this webinar, and I'd like to thank Matt Reistetter from Microbiology International to make the connections and reaching out to me to connect me with Don Whitley. And I'm happy to take questions. Thank you.